Can you see the slides, please?
We can.
Thank you.
Okay, well, we will get started. Well done everybody for making it. As said, particular shout out to Sharon, who is up ridiculously early in the morning as far as I'm concerned. We're going to kick off with Raj, who's working at the University of Sheffield with Alex Fletcher, and is doing some lovely things about planar polarity, but with cell scale models. So I don't know if Raj, you're going to move up to your sort of whole tissue models, but there's definitely a lot of really interested in detailed modeling at the cell scale that he's going to be telling us about. So yeah, take it away.
Yeah. Thank you, everyone. Good morning. So today I'll talk about the planar cell polarity. So in multicellular organism, most of the time the cells are oriented in the same direction. So, and this polarization of cells on the plane of the tissue is called planar cell polarity, and mostly this planar cell polarity is observed in epithelial tissue, which form the outer surface of the organisms, and they are manifested by hair-like structures. So what we see here in Drosophila wing and leg, the hairs in Drosophila wing and leg, and also the mouse inner ear cochlea, the hair are oriented in the same direction in this schema, in this image. Now, abnormalities in this planar cell polarity leads to like embryonic development defects such as neural tube closure defects, heart defects, even the deafness. And this polar tissue patterning depends on the self-organization, which emerge from the dynamic and iterative interaction between components at cellular level and also in molecular and tissue level. So we have today I'll focus only on the core planar polarity in developing Drosophila fibrin, which is ideal for study because it takes only few hours to develop the pattern. For that reason, we can live image the pattern to study the behavior at the cell scale, tissue scale, and even the molecular scale. So another important reason to choose this Drosophila fibrin is the molecular components are well known. So there are 6 molecules, if you can see in the schematics, which actually involved in this core planar polarity, namely the frizzled and strabismus, and there's flamingo and the disabled Diego, and the last one is Picard. So these molecules actually asymmetrically localized on the cell junction. So here in the schematic, I have shown that the frizzled molecule in green color on the distal side of the cell, and strabismus molecule in orange color in the proximal side of the cell. So they are separated, they're polarized at the like molecular level and in the cell scale. So to understand this planar cell polarity, many models have been developed in like it has been studied, and so these models actually captures with various feedback mechanism. These models captures the polarization. So what is the need of some new model? So before going to that, I'll talk about the earlier studies where actually these molecules form complex, and the complex with same orientation are stabilized, and complex with different orientation, they destabilize each other. So with these assumptions, one can actually get sorting of complexes at the local level, like local level on the junction. But to get the polarity at the cellular level or the tissue level, this is not sufficient. So when people realize that, so they introduce the global feedback. So the global queue, which is the some external field. So how this global queue works? So it actually creates some imbalance of result molecule on the distal side of the cell. And then this imbalance, this initial bias in the system is amplified by the local feedback mechanism, which I just explained in this slide. So with this amplification of initial bias, one can actually get polarity at the tissue level. But people in few experiment, it has been shown that wind morphogen gradient might serve as the global cue, but recent experiments have shown that this wind gradient is not necessary for the normal planar cell polarity. So then we are asking these questions: is a global cue is essential for cell and tissue scale polarization, or any, like, instead of global cue, some non-local feedback mechanism is sufficient for cell scale polarity? And is this mechanism is capable for generating the tissue scale polarity? So these are the questions we're asking. So we actually so here I talk about the DCO model we have developed. This model, in this model, we have realized that from the experiments, obviously, that in addition to the earlier molecules like the frizzled, stable mass, prickled, and disabled, we need some other molecule, and in experimentally we have found that the DCO molecule is very crucial to this polarization at cell scale. So we have studied the mass conserved reaction diffusion network of these molecules, where these molecules actually these molecules move from cytoplasm to the membrane, and they undergo turnover on the membrane. So by this way, there is no loss or gain of the proteins. So this is mass conserved system, and we are solving. And this model is actually developed in close collaboration with experiments, where it has been seen that this DCO molecule stabilize disabled molecule, and it destabilize stable molecule. So this experiment is currently going on, and now we already knew that disabled stabilized result and prickle stabilized stable mass. So given this information, we actually we actually solved this reaction diffusion system in a 1D in with periodic boundary condition. So here I have in the schematic I have shown that the target cell which is labeled by 1, and the all other neighboring cells is labeled by 2. And the molecule from these two cells, molecule from cell 1 and the neighboring cell, they form some complexes C1 and C2. Like C1 is the result from cell 1 and stable from cell 2, and C2 is like in this side where result from cell 2 and establishment from cell 1. So they are like in the C1 is in the distal side, and C2 in the proximal side. I have shown in the schematic. So how with this simple feedback mechanism can we actually get this cell scale polarization, that I'll talk. So the key parameters in this simulation are the destabilization parameters of the protein at the membrane, that will come later. So the results are like we see multiple peaks of these molecules, and they are separated at the cell scale. So the solid line is the C1 complex, and the dotted line is the C2 complex. So here you see that C1 complex, they actually are localized in one place, and C2 complex in the another place. So please remember that this is a periodic boundary condition. So these molecules are separated, and there is multiple peaks that actually signifies the formation of puncta, which is also important. And we also see that this prickle and disabled molecule in the prickle and disabled mutant, they show some uniform distribution, which we see in the experiment also. And our prediction is that, and we also see that interaction with neighbor cell is important in order to develop this polarity. So now I'll go into the details of these results. So one is the cell scale polarization. What we got is the cell scale polarization without global Q. That means from the random initial condition, there is no initial bias, because all the previous model, there it has been assumed that to get the polarization, we need some initial bias in the system, and that bias will be amplified by the feedback mechanism. But here we got the cell scale polarization without any initial bias. And we realized that the DCO diffusion is actually crucial. It sets the cell length scale of this separation. So to verify that, we change the DCO diffusion coefficient. The And we saw that the moderate DCO diffusion coefficient helps to polarize the system. If the DCO diffusion coefficient is very low or very high, then this complex, they don't separate on the membrane. So that is one important thing. So our hypothesis is, so this DCO molecule actually helps to non-interactly. So DCO molecule actually mediates some non-local interaction between the frizzled and stable mass molecule. By this way, they separate the complex. So this non-local interaction is important. And another important thing is in single cell experiments, we see this punctate formation of this complex. We don't see continuous density of C1 or continuous density of C2. We don't see continuous density of molecules. So we see some punctate clusters of these molecules. And in our model, actually, we are able to peak that, we are able to recapitulate that. So in terms of multiple peaks, so how we get that? Because the pinning of the frizzled by disabled and pinning of the strabismus by prickled, that actually in our model leads to this puncta formation. And this somehow explains that why we need so many molecules in the core pathway. Because we need these cluster formations for various reasons for biological functions, and to get this clustering of this molecule with the cell scale polarity, we need these multiple molecules instead of just two molecules like frizzled or strabismus. So the other thing is as I have mentioned, that these parameters, the stabilization or destabilization destabilization of the proteins on the membrane is important. So we have seen, so here is the phase diagram in the mu and tau plane. So tau is the frizzled stabilization on the membrane, and mu is the stable mass destabilization on the membrane. So if I increase tau, that stabilize result. If I increase mu, that destabilize stable mass. So now what we see, the yellow region is the polar puncta region, where they form puncta and they are separated at the cell scale. But we see the other, which actually is interesting, but we see the other phases also, like the homogeneous phase in some parameter space, and some puncta but not separated at the cell level. And another thing is like the polar behavior, where they are separated at the cell scale, but they don't show the multiple peaks. So this is the blue region. So if we decrease the value of tau, then we actually go from this yellow region to this blue region. That means if we decrease the pinning then these multiple peaks, they coalesces and form this one single peak. And most of it is interesting in mathematical studies of the reaction diffusion system also, because in reaction diffusion system, at least in two component, we don't see multiple peaks. Mostly these peaks coalesces in steady state and they form one single peak. So in that term also, it is interesting to study mathematically. So these are the results, and our conclusion is that our model recapitulates the cell scale polarization without global cube, without any initial bias from random initial condition, and DCO diffusion actually sets the length scale of the separation of these molecules. And our model actually recapitulates the puncta formation in wild type and also in mutant cells. And interaction with the neighbor cells is important for correct polarization. So future work is we are extending this 1D model into 2D using the chest software what we have. Yeah. Thank you for giving this opportunity and for your and so this work has been done in Fletcher group in collaboration with Stutt Lab. Mostly the experiments experiments here I was talking, they are done by Hong Shao and Sam Warrington, and I'm working with Jack, one other postdoc here, to develop this 2D model. Yeah, thanks.
Okay, lovely. So do we have questions? You can either just, we're a small enough number, you can either just shout them out or pop them in the chat. What does anybody have anything they'd like to ask about? Al, go for it.
So that was a really nice talk, Raj. So I had a question about the location of your puncta in your, if you go back to one of those pictures. So yeah, either the data or the simulation. So I was wondering first, so it's two parts. What controls kind of, does it depend on the initial condition where those kind of sharp peaks emerge? Is that, and
so this puncta actually formation, it depends on the initial condition, but their separation is the emerging behavior that is not coming from the initial condition, because from the initial condition, this C1 complex they are mixed. This could be like this if there is no, if only the amplification of initial condition is there, then it would look like this. But if this DCO mechanism is there, then this C1 and C2, the solid line and the dotted line, they will be separated at the cell scale. Of course, their amplification, if I change the initial condition, the behavior will look same, but their positioning might be different. Thank you.
Okay. And then my other question is, because it looks like really rich biological data that you were showing on the slide before this one. And I was wondering, from that type of data, is it possible to get like a distribution? Because it looked like the black images, there were like 3 black images of biological data. Yeah, this. Yes. Yeah, is it, so those white blobs are, my impression, or those are the puncta. Is it possible to get like a distribution of how far apart they are and how big they are, so that you can then see if your model is matching that distribution?
Structure analysis we are not doing. I'm actually analyzing this data also. So here mostly the statistics we are looking at, like how this cluster size is varying for different proteins, and like their density is varying. So, but yeah, so their structure analysis we are not looking at it right now, but that will be very interesting thing, because for different molecules their structure is very different. So earlier actually in models, so there is a scope, like mostly the experiments in this planar polarity systems where confocal microscopy, where like you see continuous distribution of molecules, but this single molecule imaging actually enable us to see the different structure of these molecules, at least like the transmembrane molecules and the cytoplasmic molecule, they look very different on the membrane, on the puncta. So it would be interesting to look at those structures. Yeah.
Sure, thank you.
Eleni, you had a sort of symbol up. Did you have a question? No, okay. Okay, Aditya. Aditya, go for it. Oh, we have a hand up. Do you want to talk?
Hello, I can't hear.
We can't hear you, so if you are talking, we can't hear the question. If you just want to type it in the chat, one of us will read it out for you. Oh, okay, maybe they'll come back or come back in with the question. Okay, I'll just have one very quick question, then we might move on. So
Yeah.
you were saying that one of your key conclusions was the influence of the cells either side.
Yes, the
So
cell
the bone
interaction
stuff.
is important.
So could you sort of elaborate on that and show where, because I think I missed their influence in this.
Yeah, so these molecules, the fragile and stable molecule, they actually so in our model we have assumed that there is abundant of flamingo molecule,
There is
which
one?
we have assumed that there is abundant of flamingo molecule. So first, what happens? This flamingo molecule, they form the homodimer. Then the frizzle comes, join flamingo in one side, and stabulus comes, join the other side. And this disabled and Diego joins frizzle, and prickle joins stabulus. That was earlier known. So now in our model, we have assumed there's this flamingo homodimer. So this cell-cell interaction is through the flamingo homodimer. Then we have assumed that there is abundance of flamingo molecule, and frizzled and stable mass, when they come to the membrane, they actually form bond and form complex. Now, without this complex formation, we don't see any polarization. So that's why our conclusion is that this bond formation is important, because this bond formation actually helps to the separation, like it actually this complex formation helps to separate these complexes at the cell level. If I
Okay,
Yeah.
okay, lovely. Right, so shall we, if you stop sharing?
So Aditya asked the question in the chat. Should I
perfect.
read it? Okay, so the question was, how are the initial conditions and boundary conditions set up for this model simulation?
Yes, so as I was talking about that, we are actually doing this 1D simulation with random initial conditions, like some random density, just and the boundary condition is periodic boundary condition, like so this cell 1 interacts with its neighbor cell, and we are doing our simulation in this around this blue line, which is like circle and circle. So it's a periodic boundary.
So when you say you have a random density, I mean, presumably it's a continuous function, or what do you mean by random density?
Yeah, it's a continuous, like some mean value plus some random fluctuations in the density.
Okay,
Around
so
that mean value.
where does the mean, so is it a uniform mean then with
Yeah,
fluctuations?
the uniform mean, and there is then some additional
Some
fluctuations.
Gaussian noise on top of that,
Yes, yes.
basically.
Yeah. And that mean value is actually we are taking from this single cell experiments, where each pixels actually denotes each molecule. So if we count the pixels, we know how many number of molecules is there, and that ratio we are taking at the initial condition, the mean number of molecules.
Okay, lovely. Right, any more questions, anybody? No, we're looking good. Okay, right, Eleni, Raj, do you want to stop?
Yeah. Thank you so much.
sharing?
I have a small question.
Cool.
Yeah,
The
please.
protein movement within the cell, if the proteins are moving on mass, do they induce cell movement?
Cell movement actually, so this whole planar cell polarity has been seen in the developing tissue. So these cells are actually moving and there is division of cells, everything is happening. So in that background, we see this development of this planar polarity. So in this 1D model, it is not possible to to have those like cell division or like the stress on the tension coming from the hinge contraction of the, so this is actually in the drosophila wing, so the hinge contraction is going on, that's why the flow in the tissue is there. So that's why we are extending our model into 2D, so where we'll study this planar cell polarity in presence of this hinge contraction and cell division both. Yeah, so I have not done this.
Okay, that's exciting because if I imagine for a minute
Yes,
that early morning that the cell is a bubble
yes.
and protein is many bubbles within the bubble, so
Mhm.
the whole bubble is moving.
Yeah, so there is the movement. So then how that change these cell divisions or the flow in the flow of the cell, how that will affect the planar cell polarity, that is actually has not been studied much, and this is quite interesting to look at. But given like our model where we are looking at only one cell, so it is not possible to capture that. But it's an interesting thing to look at, of course. Yeah,
Absolutely. Thank you so much.
yeah.
Lovely. So, Eleni, do you want to share?
Oops, sorry.
Okay.
Is showing up okay?
Yes, perfect. Are you ready to go?
Yes, I am.
Okay, right. So we're delighted now to have our next talk, which is about now something a little bit, related, but rheology and rupture dynamics in epithelial monolayers. So Eleni is actually a first-year PhD student in Cambridge, and we're delighted that she's going to be sharing what she's working on. She's working she's got collaborations across multiple universities here, which is also always very interesting to see. And we're all going to be really interested to hear what you've got to say. So take it away.
Thank you very much, Katerina. Good morning, everyone. My name is Eleni, and as Katerina said, I'm a first-year PhD student at the University of Cambridge, but I'm also working across UCL, and I have also collaborators, Alessandra, who's also on this call. My research, what I'm mainly interested in is trying to kind of relate nonlinear rheology events, so soft tissues being able to deform a lot more than what we would expect some other tissues to be able to deform, and kind of trying to relate that with adhesion and rupture dynamics at the molecular level. And as a bioengineer, I'm quite motivated to be able to mechanically characterize biological tissues because it's quite valuable knowledge for fundamental science, but also for bioengineering applications. So I envision this potentially being used to design medical implants or soft robotics or wearable electronics, etc. So let's dive into it. Now, under normal physiology, when everything is going well in the body, soft tissues are exceptionally good at withstanding mechanical forces. So many of our organs, such as the skin or the blood vessels or the intestine, are lined by epithelial monolayers, which are adapted in order to be able to withstand these loads and not break. However, under certain conditions, so when we have excessive external stresses or internal buildups of tension, or in some genetic abnormalities or pathologies, this can lead to tissue fragility and rupture, which can have severe consequences for health. So an example is mutations in the intermediate filaments and desmosomal proteins in epidermolysis bullosa, which is a family of diseases that leads to fragile skin and rupture, even under physiological stresses. So I hope I've convinced you that it's important to understand more about tissue fracture in the body. But despite this importance, this topic remains quite elusive in biomechanics research. And the reason behind this is that tissue fracture is a multi-scale process. And I think the previous talk kind of already introduced the idea of having different scales in biology. So in this case, tension at the tissue level is distributed to individual cells, which is then in turn distributed to the molecular connections between cells. So understanding how the load is distributed across different scales and how events that happen across different scales influence each other is challenging, but essential to get an accurate picture of the mechanism. So our model system in the lab, which is also illustrated here in the videos, is MDCK monolayers. So these are 1 cell thick, and you can also see the cross-section stained with GFP as we apply a load on it. So we can subject these monolayers to a stretch and record how they deform and also eventually how they rupture. And just by looking at these videos, we can kind of see that qualitatively it appears that there is some link between mechanical separation of cells at the cellular level and the macroscopic tissue rupture at the tissue scale. However, the exact interplay of the rheology and the stochastic molecular dynamics is difficult to decouple in order to pinpoint the exact mechanism of failure. What we do know through experimental characterization of these model layers is that they have a broad distribution of failure timescales. Additionally, we have seen evidence that this nonlinear viscoelastic response has some kind of deformation within the cell. So there's some part of the cell which accumulates plastic deformation and changes in some way under load. This accumulation of plasticity has actually been attributed to the keratin intermediate filaments. So potentially a mechanism to do this is that the filaments slowly unravel and straighten under load. And once they're straight, they can start bearing load and deform. So it's quite interesting to see that we have this strain stiffening response, but it's even more kind of maybe unexpected that it is also rate dependent. So in this final plot, you can kind of see that when we have a high rate, so when we kind of stretch quickly, the rupture is at a high tension and low deformation. So they kind of break quickly. But if we apply this deformation quite slowly, so we're kind of down here at the slow strain rate, we can deform quite a lot. So the rupture strain suddenly becomes quite big and the rupture tension is quite small. And this is an interesting result. We speculate that it might have something to do with tissue adaptation. So when you give the tissue a longer time to kind of deform and reach that state, maybe there are molecular mechanisms that allow it to adapt to this new load. We also have developed some computational models to capture the viscoelastic response and the tissue rheology at the junction dynamics. So on the left, you can see this network that we have built. It's a material model that consists of many Maxwell branches in parallel, which you can probably recognize. And then they also have this added component, which we call the slack. The point of this is to introduce a nonlinearity that we've seen in the network. And the idea is that these become load bearing at different strain thresholds. So you slowly load your network. The slack component is removed. And once it is removed, then you can start bearing a load. So the dash plot starts to extend. And this actually results in the same viscoelastic strain stiffening response that was predicted by our experiments, which is quite promising. And then we also have a model for the cell junction dynamics. So these are captured using this stochastic slip bond model, which is inspired by the work of Bell. In essence, what it is, it's just two surfaces, which represent this intercellular junction. So each of the pink sides are just two cells. And then in between, we have a population of N independent linkers. And these can exist in two states. So they can be bound or they can be unbound. All the links that are bound, so that the ones that are on, are equally share the force that we put on the junction. So the good thing is that both these models capture the behavior of our model system well. But any conclusions about tissue remodeling and adaptation are speculative at this point, because we have only validated with one set of data for a very specific loading pattern, which is a ramp loading, which is quite practical, nice to do in the lab, but it's not very physiologically relevant. So if we shift our focus to the function of epithelial tissues in vivo, we kind of observe something quite compelling. So these tissues are frequently subjected to cyclic loading. So for instance, we have the epithelium and the capillaries, which withstand the blood's pulsatile flow. We have those lining the lungs and the gut that experience oscillatory loads as well, different timescales, but still some kind of cyclic event. And we observe that this kind of cyclic loading is closer to epithelial physiology and may hold the key for us to be able to better understand these tissues' adaptation to resisting rupture, which brings me to my research question. Why do epithelial monolayers not rupture in vivo? So we hope to tackle this question by mimicking physiological conditions more closely. We hope to gain insights into the mechanisms that underpin this remarkable resilient behavior of epithelial tissues. They can sometimes, deform up to 300% without breaking, which is quite nice. And hoping to do this by exploring the interplay between cell-cell adhesions and the cytoskeletal architecture and components. So our investigation begins by examining the dynamics of cell junction rupture. So this is the model I showed you before, and we said that the bonds can be in two states. They can all be connected, or they can all be open, or somewhere in between. So initially we observed that the links can associate with an association constant rate K on. This is constant, but they can also unbind at a rate K off. So I've kind of made an analogy here with some kinetics models that we use sometimes in chemistry about how we can shift between these two states with these constants. Now, when these two rates reach equilibrium, we anticipate the junction to reach some kind of stable point. So the net number of bonds will be constant. This equilibrium state offers some kind of analytical expression that I've put here. I'm happy to go more into this later if you have any questions, but the idea is that at this state, the junction will be able to stay kind of constant in equilibrium and hold the force that you apply on it. So first, we want to characterize our model properties using this analytical solution and get a bit more insight into different regimes of how it can behave. So we start by applying a range of different tensions and find the pairs of equilibrium points for these values. Now, the fact that they are pairs might hint something about their stability. So we interrogated these points and found that actually one of them was unstable, meaning that the junction would still break, and one of them was stable. So for each value of tension here on the y-axis, you can kind of trace out this unstable point. It's usually a small number of closed bonds compared to total bonds, and then the junction will break, or you can go to kind of a higher ratio where the junction was able to be stable. As the tension values increase, we slowly see that these 2 points merge into one that we call the critical tension. And any tension above that value means that it's just too high, there's no stable point, and the junction will rupture. This allowed us to also define 2 important quantities. One is the rupture timescale and one is the recovery timescale. So rupture timescale is how long will it take for the junction to completely break under a certain load, and it's this state here where all the bonds are off. And the recovery timescale, which is how long does it take for the junction to reach this equilibrium solution, which will be defined for the specific tension. So you see here, it's some ratio of bonds that we define analytically. So the next thing is to systematically test our stochastic model for each part of the solution space. And that's exactly what we did. So we tested a range of tension values and compared the performance of the analytical model and the stochastic model. And it's in quite good agreement. So in black, you can kind of see the prediction, which is just the mathematical solution. And in blue, you can see all the different simulations with the stochastic model. So for each value of tension, you see blue dots, which represent different simulations, which we rerounded that value. And the means are plotted and compared. So the blue and the black match quite nicely. Now, for each tension level applied, because we tested a quite wide range, the tissue exhibits either a recovery or a rupture timescale. This means that for the lower values of tension, the tissue can recover or potentially remodel, while at higher tension levels, it undergoes rupture or damage. Now, if you see the x-axis, they kind of match nicely, and there's this orange dashed line in between, which we called the critical tension. So this was approached asymptotically from both sides, and we've kind of said that it's the transition or the switch, if you will, between these 2 regimes of healing and breaking. So this could potentially suggest, or an idea we had linking back to cyclic loading, is that by alternating tension levels above and below this critical tension, the system will keep oscillating between 2 states, evolving towards rupture and evolving towards healing. So this could suggest that the tissue's response is quite sensitive to the applied tension, but maybe we can find a way to keep it alive. So we investigated this again, and indeed we got a rich set of behaviors. So we picked 2 tension values, one that belonged to the rupture tensions and one to the healing tensions, and we cycled between them. Now in the top left, you can see that if our cycle period is quite long, so we spend a lot of time at the high tension, we still break. If we try to go a little bit faster, so we increase the frequency of our oscillation, the time scale of the material actually changes. So we still break, but we break a bit later. And we observed that and we called it fatigue. And then in the final case, we go even faster than that. And what you see in the final plot is that actually our material never broke, or at least for the duration of our experiment, it didn't break. And then those 3 kind of graphs correspond to these 3 points here on the graph on the right. So you've got the one that breaks pretty much instantly in the purple region. And that makes sense because the time spent at high tension was more than the fracture time scale. So it's expected to break. But then as you spend less and less time at high, the total amount might be more than the fracture time scale, but you can actually go either in this fatigue or healing regime here. And we tested that for also varying time spent at high and low tension to generalize this result to even more points. And this plot is quite useful for this, but also because this recovery time scale is quite difficult to determine experimentally. So it's easy to control how much time you want to spend high and low. It's easy to do experiments where you rupture and you kind of approximate this T star, but healing time is quite difficult to see. So we hope that if this is consistent across other parameters of the simulation, it could be used as a lookup plot to find the healing time. But it's also quite exciting to see that unlike traditional engineering materials, biological materials could potentially remodel and heal in response to oscillatory loading. So just my final slide on future directions. We want to assess our model's robustness across different parameters, and then we would like to test the adhesion dynamics and the rheology together to see how these 2 interact, because in fact, it's not just 2 rigid kind of interfaces. These are lined by cell membranes, which are highly viscoelastic and could affect the way that the load is distributed between these bonds. We also want to validate with experimental evidence, so potentially using DNA hairpins to kind of approximate quantitatively this number of bonds in our junction, but also attempt to quantify fracture time, healing time through counting these bonds. And finally, we would like to recontextualize these results and understand how they could be relevant in epithelial monolayer physiology. And with that, I would like to thank the organizers of the conference for giving me a platform to share my work, my supervisors and collaborators at Cambridge and UCL in Milan, and the Sensor CDT for funding my research. Thank you.
Okay, wonderful. Great. So do we have questions? As before, if we could either shout them out or type them in and we'll read them out, that would be absolutely fine. So does anybody have any questions? Oh yeah, Al, go for it.
That was a really nice talk, Eleni. So I had a question about, you were showing you had a time scale with some kind of your stochastic model and your deterministic model. And I was wondering, so what is the intuition behind why it seems like there's a lot more variability in the recovery timescale versus the rupture timescale?
Yeah, that's true. I think it likely has to do, so the X scales and the X axes are quite different in this plot. So on the one on the left, I've zoomed quite close to the asymptotic behavior. The reason behind this is because the simulations take much longer because it's healing time. So, you expect it to take longer to heal. And I wanted to kind of extend them as much as possible to make sure that they don't actually rupture at any point. And you can kind of see the same behavior very close to this critical gamma here on the right as well. So as you approach this value, there is a bit more randomness. I think the reason behind this could be linked to the value of N that I'm using at the moment. So my interface at the moment has 100 bonds. And actually I have a plot that might make this also a bit clearer. So here on the left, you have kind of 100 bonds very close to that critical value. So this is 0.063, where the critical value is 0.064. And you see that because of this stochasticity, even though this value is below your critical these simulations still break. However, if you use a higher number of n, the randomness is obviously kind of minimized, so there's less noise. And those are a lot more like closer to the analytical solution. If we wanted to use a tension that is kind of very far, then the stochasticity has less of an effect. So you still just kind of go around your equilibrium point.
Thank you. And so just to make sure I understand, so if you were to use, you know, in the limit of infinite number of bonds, would you be expecting to get you then? Okay.
Exactly. Yeah, you would perfectly match the analytical. Yeah.
Super. Thank you.
Thank you.
Do we have more questions from someone? Anything?
I can ask a
Yep, Maria?
again, I'm not very good in this field, but I'm just wondering, you talk about elasticity. Does that play a role, and how is it? I didn't catch how it's behaving in your model.
Yeah, so might go back to the experimental results because I think it's nicer to maybe use that. Or even this graph is quite nice, I think. So this is tension versus strain. So in normal stress strain, if you had an elastic material, you would expect this to be linear. So you can recover the deformation perfectly if you're elastic. But the interesting thing with these materials is that they're viscoelastic, meaning that they have a combination of elastic properties and viscous properties. And the viscous side of things make them accumulate this plastic deformation, so permanent deformation that you can't recover anymore, which is why these plots aren't kind of like just straight lines, but they have this curved behavior. And I think that's the part which makes them kind of exciting, because understanding how this plasticity might be linked to remodeling in a way that allows them, you know, in future to resist this load better or become tougher, I think is like really exciting. Does that make sense?
Yeah, and I guess for me, is it like, how is it captured in the model? Like, how do you capture this viscoelastic behavior? Is it just like the combination of two different parameters that you're sort of playing with? Sorry,
Exactly. Yeah.
Or
No, definitely. I think
basic terms.
no, I think it's definitely, it makes sense. So in the model here on the left, which is the network. So the thing I mentioned, which was a Maxwell model, is basically putting a spring in series with a dashpot. And the spring is the elastic component. So spring is something that can fully recover. A dashpot is something that is the viscous component. So it accumulates plastic deformation. So let's say we had this system that we now have stretched to a certain point. If we were to kind of put it back, the dashpot would stay at the new position. And that shows you that, you know, you've accumulated some plastic deformation in there. And these are kind of networks like this you can use to approximate what materials would behave like, whether they're elastic or viscoelastic, or, you know, you have a lot of things in parallel or a lot of things in series. It's a bit like Lego, so you can put them together and kind of approximate how your material would behave.
That was great. It was a really great talk. Thank you.
Thanks very much.
Any other questions? Just
So
another.
there-
Sorry, I
Sorry.
just need to quickly just point out to everyone that Diana is not going to be talking. So if anybody has logged on and is starting to worry about where she is, unfortunately that talk has had to be cancelled. Okay, great. Al, you have a question or you have a comment?
There is
No,
a
Alessandra,
question, yeah, there is a question in the chat, so I'm gonna read that out from Adita. So the question is, in the model, do you consider any heterogeneity in the types of bonds? And then the second question is also, is there any distribution of stress due to layering of cells?
Okay, really, really good questions. So for the first one, at the moment, all of them are slip bonds, which means that they become more kind of fragile the higher the load that they're bearing. We are able to also model catch bonds, but we haven't actually done any simulations with those yet. And ideally, we would like to maybe distribute slip and catch bonds in a way that approximates what really happens at the junction. And then the second question, I think, was about layering. So at the moment, we're just trying to keep it as simple as possible. We know that in real life, you know, for instance, in skin tissue, you have stratified epithelia. So it would be very interesting to start kind of putting more layers to the problem. But we're kind of just starting in the simplest case.
Okay, could I ask a question? While we wait for the next talk, we've got a bit of time anyway. So the equation that you showed, which I think is on the next slide, which was the sort of bell, no, it's not probably that one, yes. So N is this, in this case, is what?
So little n
Little
is
n.
the number of connected bonds, and capital N is the
Okay.
total bonds.
But is this the equation that you're simulating then, or were you doing it for each individual bond itself when you did the stochastic? I mean, how does this equation relate to the graphs we were seeing?
Yeah, yeah. So basically, you kind of want to say that all the bonds that are off have a probability of going on. So you multiply the probability of binding by the number of bonds that are off, which is 1, which is your total probability, minus the fraction of bonds that are connected.
Mhm.
And then the rest of the bonds have a probability of opening, which is given
Yeah.
by this equation. So this equilibrium suggests, and this is the equation that I've used to model this graph here. So
Okay.
to kind of link, what ratio of bonds would correspond to each of these tension values, and are these points truly an equilibrium point, or is there something else going on?
Mhm. Mhm. And then when you do the simulations, what are you simulating?
So I'm using a Monte Carlo like stochastic process with these two equations. So each bond.
Okay, so, but you're using the probability of binding and unbinding based on those two things.
Exactly.
For individual bond, in effect.
Exactly. Yeah.
Okay, right. So that's great. So when you say equilibrium, what do you mean? Are you looking at dNdt is 0, or I mean,
Yeah.
so what's the equilibrium? Okay, perfect. So then you do this sort of basically stability analysis, which is on the next slide, for the entire ensemble. And then when you're doing the simulations, you're looking at the individual bonds in a stochastic process, in effect.
Exactly. Yeah, and you can track the individual bonds and kind of see like how close you are to this ratio. If you're above or below, what would happen?
Yeah. Okay. Lovely. Great. Okay. So I understand what's going on there, which is lovely, and it is a lovely model.
Thank you.
Does anybody else have any other?
Yes,
Yeah.
can we learn anything or consider some knowledge from wound healing?
Yeah, that's a great question. I like looking for applications for my work very much. And I know there are some people in Cambridge working on that as well. So wound healing can to a certain extent be linked to this healing of the monolayers or remodeling. So I don't know too much about it yet, but it's definitely something that I would like to explore. Yeah.
So your model is like within the cell, right?
Yeah, so in the monolayer,
focusing on processes within the cell. But when there is a rupture, is it localized enough that we can talk within the cell about some kind of environment, micro or nano environment within the cell that may mobilize and help the healing? Actually, I'm repeating my question, actually.
yeah, so I think that kind of links to the multi-scale level of the problem. So in some cases, between the individual cells in the junction, you can have mutations that affect, for example, your cadherins or some of the desmosomes or some of the components that keep the cells together. And if those are mutated or have some kind of pathology, then that could lead to your skin, the skin condition. So it does translate from the cell level to the tissue level. And it's exactly that mechanism that we're trying to characterize.
Okay, great. Thank you.
Thank you. Katerina, I think you are muted.
Yes. I was just about to say, do we have more questions? But I think not. In which case, we will just take a sort of short 5-minute break, give ourselves time to clear our heads. We can thank all of our speakers again. So however, that was really, really wonderful. And I suggest we all just sort of take 5 minutes to clear our heads, and then we'll start with the final plenaries of this session.
Maria, is this coffee or tea?
Coffee. I got it delivered. You just can't see my. It is.
It was
too
it's good.
early.
It was great sadness today. I got to campus and I realized that the coffee shop didn't open until 7 a.m. It's like, no. So I will get coffee after I teach.
Yeah, no, that's my favorite thing, in the morning. That's kind
Yeah,
of
absolutely. It looks like Katerina's here.
that's great. I do
Okay.
want to say like one more time, Eleni and Raji, you both did wonderful. I enjoyed both of your talks. Hi, Katerina.
Hi, Katerina.
Thank you. Thank you.
Welcome. Yeah, those were a lot of questions too for both your talks. I think that highlights how good they were.
Hi, we're just having a short break to clear our heads before we get going. We encourage everyone to sort of maybe stand up and move around a bit. It's been sitting here in front of the screen for an hour. How are you, Katerina? I haven't seen you in ages.
Yeah, we haven't, as we
I
said.
know.
SMB 2020 online. Yeah.
Yeah, something like that.
Yeah. And then I was supposed to come to Sari and my food injury and
I
getting
know.
it was in the middle
You
of it.
bailed on us. I was looking forward to it very much.
Yeah, I'm still struggling with that actually. I need to do much more intensive physiotherapy apparently, I learned yesterday. Anyway, so not much traveling. I managed to come to Cyprus with special assistance, but I'm staying here until May. I don't know who is hearing me now because I'm saying too much personal info.
Everybody is hearing you. We're
Oh,
all
I'm sorry.
together. You are in the general session. But we're all
Anyway,
very sympathetic.
I saw the tweet about excellent work from Eleni and the talk. Sorry that I couldn't join earlier. I had another meeting. I could not move in the end. Hi, Eleni.
Hey.
Well, you are recording everything, right? So
Yeah, so you can come back and you'll see it, which is wonderful. Yeah, indeed, it was a very nice sort of mechanics and viscoelasticity and stuff. So right down our
okay.
sort of interest, Katerina and I.
I definitely have to watch that.
Yeah. But,
so Eleni, you are a PhD student or a postdoc?
I'm a PhD student in Cambridge and also will work a bit in UCL.
Maria, we met before.
Yes, I'm looking forward to seeing you today too. No, it's great.
Alexandria,
it's
you, like I see some people I know also from the.
And
great. Well, thank you for organizing this. It's great, but I thought you wanted to go on a break, right? Since
I've done my wriggling around. It's fine. Everybody else is.
You just stretched a bit, and now everything is back on track. Okay.
Anyway, I'm tough. I can make my way through.
I know you're tough, but maybe this is not the message we want to get through to the younger people, right? It's like we're going despite everything.
Katerina.
Relaxation and
Maybe Karina only looks like she's sitting. She's actually squatting. But I wanted to say, Katerina, I'm gonna have to leave your talk after 15 minutes to go teach my class, but I will look forward to
Okay.
the first part.
That's fine. You will see Thomas part then. Thomas is
Okay.
joining in a few minutes because he has teaching.
Yeah.
So
I was saying before this started that I was telling my guides that, you know, I had to finish my teaching early in order to set up this session. And I was really disappointed. I had no tears. I had none of my students saying, no, we want more of our lecture. They were all just kind of like, yeah, fine. So, you know, what does that say about me?
I like Maria's. Oh,
sorry.
I was saying I like Maria brought up the point of how much it actually costs, like 1 hour of instruction. If you do that by the cost of the university, how much it actually is worth.
How much is it?
I have no idea for us.
Well, it depends. I was just dividing it by tuition. Like it depends on what your tuition.
Right. Yes. Yes.
Yeah, it's a bit high on an hourly base. If you think about how many, classes you have in one semester. I mean, of course, but if you just like do a rough, it's a big chunk. And
yeah.
So Katerina, are you going to go first? Do you have slides? Should we just check that they?
Yes, I have slides, but we coordinated with Thomas. You want me to start before he
No,
arrives?
I was just checking what the plan is so that we can be ready.
He's starting because basically we did some kind of chronological. Oh, I don't want to give it away.
No, don't give
Thomas arrives. He starts.
Okay, we wait for Thomas anyway, and then
we're waiting for him. So
and for everybody who's listening, it's worth the wait. I think I enjoyed every talk Katerina and Thomas have given separately or together, and I just mean like it is worth the wait. So
you're too kind. I'm gonna blush now.
I'm already blushing. Can't you see? I'm always red.
Oh dear.
No, it's the truth. I'm not saying.
Yeah.
Oh, thank you. But are you collecting your drawings into an edition or something, Maria, or what are you doing in that direction?
Oh, my scribbles? Nothing.
You just keep generating one by one, because it would be great to have a collection of those, isn't it?
Yeah, I could always share them, but I always wonder, do you understand them? Like, aren't I scribbling too much? So it is.
No, I think they're beautiful. They're always beautiful and fantastic. And the other thing I think about them, which is it would be a wonderful idea which Katerina's come up with, because I think the other thing they always illustrate is the breadth of any piece of work, how much there is to it, how much scope you bring out, how much scope is covered by people, which I think is really a lovely message for people to see, that it's not this little tiny equation and very sort of narrow thing.
I wonder if you could put them in like Bulletin of Mathematical Biology as a article in that. It's like I feel like the community would respond well to that.
Absolutely. I think so as well, yeah.
We can put them on our website. I mean, they already have the CDEV stuff if you like to do that.
Yeah.
I mean, that'd be lovely too, but I feel like they could be in a published form too.
Yeah.
I feel like I mean, I'm a scribbler, and when I was learning to teach anatomy, so one of my professors and I during my grad school, we were teaching anatomy, and that's where I really realized that that's my number one way to learn and memorize fast. Like if I don't want to go back to it, I write it down. If I type it, it might as well not have been written. Like, and I don't know why. Maybe the keyboard and we haven't linked, or I didn't evolve early enough with the keyboard, but typing something doesn't mean it goes in my brain. But if I take a pencil and I start doodling, it's a completely different story for me. And once I was able to get an iPad, that was kind of where I went right away.
But I mean, that's a skill very few people have, like the speed with which you are generating those. I mean, I was very impressed.
Yeah. Do you mind if I just share one of them? I've just picked something off your Twitter page so that anybody who's here knows what we're talking about. I think that would be quite nice.
Yeah, that'd be good.
Oh, yes.
Yeah,
absolutely.
because we've all just sort of know.
Yes,
Maria-
oh yes, maybe other people don't know about Maria's story.
Yeah, so Maria has a really wonderful way of illustrating work and talks. So when she is taking notes from a talk of some sort, I have no idea which one this was, I've just taken it off of Twitter, but it's very beautiful how the different information can be displayed with sort of pictures and words and colours. I mean, this is one of the more plain ones. Sometimes they're really, really sort of the images are beautiful, and Maria's a very, very talented artist. So that's what we're talking about here.
Thank you. And you've managed to make me more red.
But the thing is, Maria, you can make money out of it. I just clicked that. Actually, we used an illustrator for 2 events, and we're gonna show something in the talk. And she charged us £800 for just a single day of an event, just to be there and transcribe into, well, similar things you are doing. So it goes at a very high rate, actually. And one worry we had was that would she understand what we are saying? So I think people that are also scientifically literate, which I know is an understatement for you, will have a headway. If you have both the science and the skills and drawing skills, it seems that there is an growing interest, and she's very busy, and we had to find the specific day that she can. So anyway, let's discuss further. I mean, to tell you, because we tried quite a bit to find somebody to do that. So it's interesting. I hope we are still within the theme of the conference.
Yeah. I think actually we definitely are. I mean, I think it's also about sharing best practice and about different ways of working, different ways of learning, different ways of presenting information. And I think it's really, really helpful to discuss this and show there's not just one way. You sit there, you write your notes, and all the rest of it.
Absolutely. Yeah.
We might do a couple of introductions also, since we're all here. Because I don't know, Sharon, do you know Katerina? Okay. Katerina.
Here he is. Here's Thomas. So why don't we
Yeah, very
do
good.
an
Yeah.
introduction to people now to everybody.
Very good.
Thomas
Yeah.
is here.
Yeah, I think we should get started then.
Yeah,
I agree with you, Karina. Yeah.
Okay. Hi, Thomas.
Oh, hello.
Hi.
How you doing?
Are you completely breathless and sweating, or are you okay?
I mean, that's my natural state.
Okay. Right.
Thank
Okay,
you
everybody.
for delaying slightly for me
No,
to
no,
arrive.
no, no,
Just-
we've been having a lovely chat about this and that, and that's kind of the point of the meeting anyway, is to just have a relaxed chat with some people. So we're going to get going. So we're delighted to have Thomas and Katerina here. So I've known Katerina since we were PhD students together, and she was doing stuff on sonic booms, and I was doing stuff on shallow water. So we've both come quite a long way. So since then, I know Katerina's had a very interesting set of experiences working in terms of industrial and knowledge transfer, moving into Cyprus, coming back to Cardiff, and just an enormous range of experiences and some absolutely awesome work now across a range of applications in mathematical biology, cell signaling, and this new initiative on IVF. Thomas, those of us who've heard him talk know he's an excellent science communicator, and as well as the excellent science, has been doing great things with zombies and all sorts of things. So I'm sure we're all going to be hearing that. There was also bee flight or something recently. Anybody who's heard both of these people talk know it's going to be an excellent, and I'm building it up now, putting pressure on.
Yes, we rise
We're
to
gonna have a
the
really fun time for the next period, and so please do put questions in the chat at any point if you want to ask questions, and when we find a suitable moment to interrupt, we can ask them, and there'll be questions at the end. So away you go.
Well, thank you very much. Katerina, do you want to do any more introduction for yourself? Or?
Well, I mean, Karina did such a great job. Again, blushing here. So thank you, Karina. And thank you, reconnected after many years, which is great. Thank you also to the organizers for inviting us. And I I mean, just for me, just more formally, I'm a reader in the School of Mathematics and then director for impact and engagement there. And yeah, Thomas, would you like to say something more?
So yeah, I agree with thanks. I mean, what we've been asked to do with this talk is not just sort of a straight research talk. So I will be talking a little bit about the work I did, but what we've been asked to do is sort of expand on the twists and turns that this project took. Because this story starts 7 years ago. I've been in Cardiff now, coming up to 7 years, and that's where it really started. And I no idea back then when it started that it would lead here. That we have a thing called a GW4 grant, where Cardiff University, Bristol, Bath, and Exeter, all those universities put some money into a pot, and you bid to do a network connecting those 4 universities on an idea. And we bid and were successful in generating funds to generate an IVF network, looking at fertility from a variety of different points. I mean, that's what always is important to me as a mathematician, to talk to someone who isn't a mathematician. I know mathematics, you know mathematics. We can, I can come to mathematical conferences and find the new big idea, which will be just machine learning. I don't care about machine learning. There are some things that excite me and some things that don't. Machine learning isn't one of them, I'm afraid. But so what I've, as my career has evolved, my trajectory, what I tend to go to now is, to give you an example, I think it was mentioned, I've been doing a bit of bat work. So in September, I'm going to the National Bat Conference in the UK, because I've done some work that I want them to understand. I've been working with ecologists, so I've always been kept on the right track, but I want to get it out to that audience. I want to get it out to that field, so that they can hopefully use it. We've always tried to make the work useful and usable, but also B, to see if there's any more work out there that I can do. As an applied mathematician, you're never far away from an interesting question. But anyway, let's get on to IVF rather than the bats. So IVF, my particular part of this is going to be on the idea of where's the PIP2. PIP2 I'll talk a little bit about in a second. It's a protein. But it was a really nice piece of work that happened when I just joined Cardiff, because a lot of the work was done for me and I inherited a lot of it. So just to sort of hit the ground running, really nice piece of work, which hopefully you'll see worked quite well. But how did it start? Well, this is down to a collaboration with a biologist, Professor Carl Swan. And I'd met him in Oxford previously. One of my supervisors had worked with him. So, he got me to chat with him. But nothing came of it, and that's fine. As a mathematical biologist, as an applied mathematician, you invest a lot of time in tea breaks. I always love to have a nice tea break with someone to see what they're working on. And we had a nice chat. Nothing came of it, absolutely fine. But when I moved to Cardiff, I went and made a nuisance of myself in the biology department. I did some talks at various groups they had and said, okay, here's what I can do. How can I help you? Carl came up to me and said, okay, I've got a question here that I think maths could help me understand. And it's all about where this PIP2 is. So what is PIP2? Let's get into actually the sort of maths question, the biological question, and then the maths question. During fertility, one of the first things that happens is a calcium wave spreads over the egg. That's sort of an initiator that the sperm comes along, hits the egg, and a nice spread of calcium hits the cell. Now, these are xenopus oocytes. They're nice, big, bright, shiny ones. These are the ones that work well. The ones that Carl uses are, he works on them, xenopus sometimes, but the ones we for this paper was mouse eggs. But I just love this image, so I use it. But what they do is they fertilize the egg, and then they take these fluorescent readings, and over time, you can see you get these nice, lovely, lovely oscillations occurring over a prolonged amount of time throughout the egg. This has been researched a lot. So, I'm not going to revolutionize anything here or reinvent the wheel. But the question from Carl was to do with this PIP2, this protein. Because here was the dogma. You have your egg, and the sperm comes along and releases PLC zeta. Whatever, I'm the mathematician here. I just nod and agree with 3-letter acronyms. I know mathematicians do this all the time. We're very bad at nomenclature. Biologists are worse. I don't know if you've ever seen, they usually start off with a big network diagram of genes and cells, and they've all got 3-letter names. I don't know how anyone keeps it all in their mind, but I'm impressed. So anyway, the sperm has this this protein, this PLC zeterin. It hits the egg and it releases the PLC zeta. And what it was thought is that then, and I will read this directly from the paper because I can never remember it, PLC zeta hydrolyzes its substrate PIP2 to produce the calcium-releasing messenger IP3. Okay, so PLC zeta in the sperm hits the membrane, that activates the PIP2, that then activates the IP3 in the cell, and then that kicks off calcium-induced calcium release. So we have some calcium in the cell, we have some calcium in the endoplasmic reticulum, and that oscillates. So we get this wave traveling across the cell, and then that can then feed back into the PIP2. So the PIP2 is essentially the gatekeeper of the start of the calcium cycle. And the dogma in the literature that Carl wanted to challenge was, where is that protein? Because in certain eggs, they believe it's in the membrane, but then some work has been done showing that if you remove the PIP2 in the membrane, it still works. So where is it? Now, I inherited this model. As I say, you know, they've done work previously with other collaborators, and this was the model they used. And I'll be honest, I don't like it. But this is one of these points that comes to, I want to sort of expand upon in terms of collaboration. You don't have to like it. You've got to think about who is doing the work here. I was being a hired gun, essentially, for the biologists. So they want me to use this model. I may not agree with it, but that's what they want to use. I will use it. As a mathematician, I can then go and pull it apart later. You know, I can say, oh, this is wrong, this is terrible, this shouldn't work. But that's my work. I don't want to interfere with them. They believe this model, fine. And as you'll see later, it does work. Annoyingly, it does work. But there's problems with it. And I'll highlight at least one of the problems as I go through. So we have this calcium. There's calcium in the cell. That's U. So calcium is just sort of floating around in the cell. And we have V. V is the calcium in this endoplasmic reticulum. That's sort of locked away. And then we have P, which is this PIP2. And that's what helps the communication between the U and the V. There's a couple of things about this. So what I've added then are these source terms. Where are these? Where is the U, the V, and the P interacting? So the U and the V are controlled. They can only interact in the where the endoplasmic reticulum is. Okay, so that tells you that we only get this interaction form K whenever this S is on, and it's on in the endoplasmic reticulum. So we get the conversion back and forth. PIP2, where's that? Well, this is another source term, and I'm going to be changing this throughout the talk. Sometimes I'll have it in the boundary, sometimes I'll have it throughout the cell, sometimes I'll have it localized. But that's what I'm going to control here, and it's going to be controlled in this kind of way here, where I'm going to assume there's local splodges, a grid of splodges, and the separation of these splodges is L. Increasing L separates them more. Decreasing L, if L is 0, then PIP2 is everywhere. I can also change S to just be the boundary, making just PIP in the boundary. But why don't I like this? We have this really complicated K term. And it was made to look right. What do I mean by that? Well, calcium undergoes this self-induced activation. So we have a U here. This is the U in the cytosol, and that increases K. So that means that the U in the cytosol will increase the U in the cytosol. It will draw some U from the endoplasmic reticulum. Fine. This is then multiplied by 1 minus a Hill function of U. So as U increases, that then turns this off. So you have U as a positive feedback, and then as it gets too high, it then turns itself off. Absolutely fine. I've no argue. That is what is thought to happen. But here's my problem. You can set that 1 minus Hill function to 0, and it still works. That term does nothing in this equation. It's this V term that drives it. It's the depletion of the endoplasmic reticulum that drives the oscillations, not the 1 minus U term. My anger aside, my upset, my desire to be correct aside, that is the model I inherited. That is the model I will work with. I can work later on pulling this apart. And we actually had a student, Katerina may talk a little bit later about the student who did pull this apart. But that's the setup. That's the setup. And here, you know, you've got 3 PDEs. not much to do but simulate it. And this is what you get. This is, in this case, I put pip2 everywhere. So my S is just 1. There's pip2 everywhere. I give it a kick of calcium in the top right, and we get a nice little wave flowing through the egg. Wonderful, lovely, great. What happens when I localize the pip2 to the boundary? So I say that my P only occurs in the back. It's allowed to diffuse slightly, but my P source, this S, is purely localized on the boundary. We don't get a wave. Essentially, all we get is a bright ring, but it doesn't cross the cell. Okay, so it could be the model's wrong. As I say, I've got problems with this model, but it could be that the model's wrong. It's not the right model. Fine. Now let's do a bit more simulation. Now let's separate them. So in the 2, 3, and 4 micrometers, what I'm doing is those splodges, and I'm separating them. So 2 microns apart, 3 microns apart, 4 microns apart. And what you see is, and what I would say is that I didn't parameterize this via data at all. This all the parameter values, all the equations came from previous work. When the separation of these vesicles are 2 micrometers apart, we get a nice quick, the wave crosses the cell in less than 2 seconds and it stays there. When these two regions are 3 microns apart, it's slower, but there is still a wave. When they're 4 microns apart, no wave at all. And when it's on the boundary, no wave at all. Okay? Does that make sense? Now we have some data. So what Carl was able to do was use a reagent called propanolol to reduce the amount of pip2 vesicles in the cell. So here's your wild type on the left. The red is all the pip2 in vesicles. So they know some pip2 is there, but they don't necessarily know it's active. It's sort of wrapped up in membrane. On the right, they've added this propanolol, and it removes some of these vesicles. And so that's what we're looking at. Ignore the left graph and really sort of focus on the right. In the control, the distance between the neighbors of the pip2 vesicles is around 2 microns. When you add the propanol, it goes up to 3, 4 microns. And that matches exactly what we see. In the first case, we get a wave crossing the cell. When we got to 3, 4 micrometers, the wave breaks down. And that's what you see in the data. You have a nice control cell, nicely oscillating calcium. You add your propanolol, remove some of the pip2, removes the oscillations. You take away your propanolol, brings back the oscillations. So it's a nice little package. It's a nice little piece of work. I say I can take very little credit for most of the stuff because I inherited it. I just mainly simulated. But it was just so nice that everything, as I say, I don't like the model, but there's no denying it works. So I'm going to pass on to Katerina now, but I just want to say, the conclusion for me, for the biologist, was pip2 must exist throughout the cell. There is a region with it. If it's too separated, you don't get your wave. But what happened on the way to getting this and forming this bigger IVF network? Well, firstly, as I say, my supervisor got me in contact with Carl. And then I made myself a nuisance in the biology department. They want to talk to you. Trust me, you won't be a nuisance. They have skills, you have skills. Your skills are compatible, they are complementary. As long as you're nice to them and you don't go in and say, you all suck and you need me, then they'll happily work with you. Who is the work for? This idea is you may not like what you're doing all the time, but be honest with your biologist. And if they're happy with your assumptions, this is a key thing. There are many times when I've had to really be, my cell moves because I've attached wings to it. Does that make sense, biologist? And if they're saying yes because of X, Y, and Z, then there's not really much you can do. You can stress your assumptions as much as you like, but as long as the biologist is willing to accept it and justify it, then there must be some truth. Luck. You're an academic. Luck is going to account for more of your career than most. Luck, and there's a nice phrase, luck favors the prepared mind. The more work you do, the more collaborators you have, the more handshakes you have, the better your luck will be. But this goes into what Katerina will talk about. Whilst I was doing this work and Katerina joined and we were setting up doctoral positions, we got a phone call from a local IVF clinic wanting to work with us. None of us had set that up. Pure luck. And it's built and developed into a wonderful way. And academia is the long game. As I say, this has been 7 years plus worth of work going on here. I've told you one story that's been 15 minutes. That's a good 6 months of my life. So very often, if you're sitting there and you're a younger researcher, I often saw some great talks by very senior people and thought, wow, that's incredible work. I'll never get there. They've lived a lot longer than you have, and they're cherry picking the good stuff. They'll have done a lot of rubbish as well within that. And with that, I think I will pass along to Katerina to show you all the other good stuff that we've done.
Yeah. Thank you, Thomas.
How do I stop sharing that one?
I'm sharing. Can you see my screen?
Yep, it's just come through.
You can see everything, right? Because I kind of not see everything with the bar below the Zoom, but yeah, I think you can. Great. So yeah, so as Thomas said, luck plays a role, and I think it's luck that brought us together in Cardiff. I joined 6 months after Thomas, and I was working on calcium signaling before, and then I mentioned this to Thomas, and Thomas had this paper. We're wondering, oh, let's do something together. He introduced me to Cars1, and then this phone call came. So the phone call was very weird because I've done lots of things with industry, and it's very rare that a company seeks you out, especially a clinic. But there was this embryologist that was very eager to do a PhD. And so she was looking for mass help. So she contacted an admin person in the school, not even academics. And then the next step is where the luck has to be worked on, right? Because the moment this call came through, we chatted with Thomas, said, let's go over, let's meet them. Luckily, they were right down the road, like 5 minutes walk, London Women's Clinic. So when there, we had a meeting with the embryologist and her manager, Giles Palmer, who we still talk to this day. And then another opportunity comes up with this CAS2 academia industry grants. So we said, let's go for it. The company was very eager to answer a question about choosing the best embryo. So they accepted to co-fund a PhD studentship with 1/3 of the cost. 2/3 came from this CAS2 scheme that is an EU grant that was allocated to Wales. So it was a lucky occurrence again. Things came together. We got the grant, and we recruited a team Ostler. I think I haven't done the PowerPoint mode, isn't it? That's it now. Yeah, that's what I was looking for. So so they came with this question. They said, does the growth rate of embryos tell us anything about whether a pregnancy will occur? And this is a holy grail question, right? Nobody has answered this question yet. So it is a research question. It was a research question. As I said, we got the grant. We recruited an excellent PhD student, Tim Ostler, who will be mentioned quite a few times in the next few minutes. And we started looking at various things that the company was interested in, trying also to keep them balanced with more technical maths, because it's great to work for an industrial partner funded by an industrial partner, but at the same time, we're working in a maths department, and the PhD students need to have quite a bit of maths in their thesis. So this was reflected also in the way we split the work for team. So the original strand of the work was to do with choosing the best embryo. When I come back to that, then the clinic said, oh, what about the temperature of embryos? So this led to another strand, which is the thermal modeling of embryos. And then coming back to the fact that we needed also more technical stuff, and partly because COVID happened in the middle of team's thesis and we couldn't get more data from the clinic, we went very mathematical. But then we went full circle on how this math can be applied to IVF, but also other biomedical images. So just to tell you a bit of the work we did with the clinic, but before that, yeah, there is this part that is confusing me. Sorry, it's just taking up and down. But why is this an important challenge? Why is IVF being discussed? Well, it's a huge industry. and it's also a big societal challenge because more and more people are seeking IVF. Many people cannot afford it. It also has low success rates, approximately 30%. The success rates are stagnating. That's even more problematic. They don't know why things are not improving. And each treatment, if anyone here has done IVF, is physically, mentally, and financially challenging, draining. So how can we make IVF better? That would really help many people. So coming back to the original question that Tim set out to answer with our supervision, does the embryo's growth rate correlate with pregnancy chances? He used eventually a bit of machine learning, even though we're not machine learning experts, but this allowed Tim rather to quickly delineate the various compartments the embryo has inside, because it's not a homogeneous mass, right? And these two pictures on the right tell you that basically if the embryo is growing erratically, so to speak, then it's less likely to be born. So you have lower pregnancy chances if, as you're growing, you're doing things that are like up and down a lot. However, with this metric, this is a new quantification of how this embryo growth rate is evolving. And we predicted that they would get up to 3% improvement in pregnancy rate if our metric is applied. This metric has not been applied yet, but there is more work to be done. One of them is to get more data. There's a caveat here. We only had 124 embryos. We tried very hard to get more data. Marta Sala, the original very enthusiastic embryologist, left the clinic by that time. This is one of the problems IVF industry is facing. Embryologists burn out or move to other clinics. So you keep having these people changing. So we got some embryos while she was still there. Then COVID happened as well. And now we have this result, but it's only with 124 embryos. And we estimate that you need at least a few hundred to be able to be more statistically significant. Actually, Tim did a power of test for this to find out. And I think it was about 560 embryos at least. So we also
add one more thing onto that. So
yeah.
We had this 3% that you may think, oh, that's 3%, that's nothing. But as Katerina mentioned, the success rates are around 30%. So every single percentage you can increase that by is millions of pounds, dollars saved. So 3% may sound nothing, but if we could show this as an effect, huge change.
Indeed. And then we also were asked other questions. Is the average growth rate of the embryos different between those frozen on day 5 versus day 6? So again, clinics have to make a choice here. Would we freeze the blastocyst, which is the growing embryo, after 5 days in the lab or after 6 days in the lab? And we found with the data we had again that the answer is no. It doesn't matter if it's day 5 or day 6. Then another question came about, should all embryos be frozen? And we participated in joint work with the clinic, and we went to with a poster to Fertility 2021, which is the premier conference for UK embryologists. And basically the answer to that is only those with blastocysts should be frozen. So just to show you who was involved there, as a London Women's Clinic. There was also there's a chain of clinics across the UK. So there was somebody from the London clinic, London Women's Clinic in London. And you can see here many people, some of them embryologists, we are the mathematicians. So it was the beginning of this interdisciplinary collaboration, basically. It was the first thing I think we did, got out from the work. Then we went to the thermal modeling that I mentioned earlier. I keep talking about frozen embryos. So basically what happens is that you need to freeze the embryos very rapidly in order to avoid damage. They managed to maintain about 96% of embryos. So basically when they get them out of the freezer, sometimes after years, 96% of them recover. But of course, it's an open question whether even if they recover, have they have any damage left. But they are doing well. And of course, the specifics of how they freeze and how they store are very important. So we're looking into the freezing process called vitrification. And we use simple COMSOL multiphysics. Some of you might have used it here to just solve the heat equation. But what COMSOL multiphysics can do is to actually create a replica of the freezing device on the computer, right? So that's what we did. And that led to a paper that some of you might want to look further to find out. Again, this was a poster to a biological conference. As Thomas said earlier, go to where biologists are and talk to them as well, not just do maths. And the important conclusion here is that mounting multiple embryos does not influence cooling rates during vitrification. Why is that important? I mean, it's a nice poster. I like the colors, that's why I put it. But the main message to get out of here is that it saves clinics time because you can have the protocol more flexibly, right? As these tiny little embryos are being mounted on the device, the embryologist has to spend her or his precious time to do that very carefully. So what we're telling them is that it doesn't matter. Just put them wherever you can on the device, and you can put up to 4, which is the manufacturer's device. So it saves them time, which is very precious commodity in the IVF world. And a corroboration on that, this is interesting, is that even the manufacturer of the device reposted our poster on X, well, X Twitter. So coming back now to the more technical things, which I'll try to not relay very technically though, the more mathematical aspect of Tim's work centered around an imaging algorithm called the differential dynamic microscopy, which was used to derive diffusion coefficients in other data sets. So we wanted to apply this to eggs and embryos, and to understand, without touching the eggs or embryos, so non-invasively, how healthy these eggs are. And you might ask, why is this important? Well, we want to work with healthy eggs and embryos so that we increase the IVF success rates. A key result that came out of that work is that we should combine with PIV to get the accurate flow patterns. But DNDM, if it's a system where you just need to characterize diffusion, is doing very well. And Tim actually modified DNDM to account even for more scenarios. So this leads me to the next slide. Just to give you a bit of more background, the basal speed in X, is like some background motion they have, is the one corresponding to the diffusion coefficient. And the hypothesis that Carl Swan has, the experimentalist that Thomas mentioned, is that this basal speed it directly relates to metabolic health. So in other words, diffusion coefficients tell us something about the health of embryos. So how do we do that? We use, again, the diffusion coefficient. Now the dependent variable is something else. And we apply the DNDM algorithm, which is basically having a stack of egg or embryo images or any other images. In fact, they had colloid in the original paper by Germain in 2016. You can use the fact that diffusion is isotropic to allow to work only with differences between images at subsequent points. This is the premise of DNDM. And you create the DNDM matrix, and you can get diffusion out in that way. I'm doing very fast because I think the technical details are not important. What more important is this, that Tim managed to go back to the DNDM algorithm, question the assumptions, and this is how part of our research is happening, right? We get a model. Thomas said, I didn't like that model. I used it. I produced some results that tied up with experimental data. Another way to work is that going back to the model, and we've done that in other parts of our IVF work, and question the assumptions. Are the assumptions valid for the scenario of choice? The scenario of choice here is X. They are round, right? They are not colloid data. So initially, team ran the only in that squares inside the egg, removing the boundary effect, but that was losing information. So he adapted the algorithm to account for boundaries. And indeed, there were many differences seen when the including boundaries was boundaries were included. There were other technical results I'm not gonna bore you with, but I guess the takeaway again is that you can use NDM to extract information about any biological data set. And the work that team has done with combining with advection speeds and introducing the idea of combining NDM and PIV is the one that can allow you to statistically quantify egg health. We haven't yet published that because more work needs to be done, especially the combination with the PIV. But the question to you, do you have perhaps phase contrast images you could try the modified DDM algorithm on? So we're always looking for new collaborations, and if you have images that we can work with to apply this new algorithm, it would be nice. I will just take a detour from very technical and say disseminate your work far and wide. And as we are chatting in the beginning, Thomas, before you joined, Katerina was saying there are various ways you can talk about things and there are various audiences. So for example, at some point, I don't remember how it came about, but somebody contacted me and said, would you like to write an article for Scope magazine, which is not a mathematical magazine, it's the layman or laywoman audience. And then, of course, I talked to the team, Thomas and Carl Swan and Tim Ostler, and we wrote this article together. That was about 2 years ago, Thomas, which basically is a nice way of like paragraphs that show the various challenges in IVF.
Again, it also shows you the academic timeline. So yeah, although this was written about 2 years ago, it came out spring last year, a year ago. So
oh yes, they took them a few months after they rushed us to send them the draft to get it out, but we're very happy it's out there. Spring 2023 came out, and then the other success we're very proud of as supervisors is that Tim, who's we are in the UK, may know STEM for Britain. Tim was a finalist there. And again, he had there a summary of the IVF work he has accomplished. And again, I think it's a very nice poster. So basically, coming back now to non-invasively assessing X, another thing that brings a full circle back to our discussions with Carl Swan is a big problem he's been working on for many years and directly relates to Thomas' talk, in where you're trying to find a good model to understand calcium oscillations and waves in X. And this model doesn't exist yet. Thomas already said he didn't like that model. That model has many deficiencies. So what is the actual challenge? Just to show a video to make this challenge more tangible, is that you're gonna see here, when I play this video, finally, yeah. You see, this is from PIV, particle image velocimetry, which I hadn't elaborated on before what it was. You see these flows, right? Little arrows. And you see in the panel below, calcium oscillations. These are waves, but you cut them at one location, so you just get oscillations. And then you get also, every time the calcium oscillation happens, you get a swishing motion from the egg. It's like Carl calls them spasms, which is a Greek word, but I cannot pronounce it in English, unfortunately. So this correlation is important. So calcium waves are a marker of IVF success. This was found very recently in 2018 by a clinic in, I think it was Belgium. I don't remember exactly, but anyway, it's published, it's 2018. However, to track calcium, you destroy the egg. So it's throwing the baby out with the bathwater. So what can we do? We can explore this coupling with calcium and mechanics to get to study mechanics non-invasively, going back to imaging again. And then this hopefully will lead to a new day one. That's very important because we already talked about day 5, day 6. So they are keeping those embryos in the lab for 5 days at least. So imagine how much savings they would do if they managed in day 1 to understand whether the embryo is the one to be implanted. So this is Carl's big vision that this should be achieved, but the maths is missing. So Thomas took the first step, and then when we started working with Thomas, we supervised 3-4 other students, their names are below, to progress this model. And we are here 7 years later. We still haven't found that good model for calcium signaling in X, but we have made progress. What progress have we made? Just a very short detour. Calcium is very important and is everywhere. This is what this slide should say. There are deterministic calcium signaling models. So we went with that. Thomas' model was deterministic too. And the very big improvement we have made and made Carl very happy was that there were these experimental data back from 1998, and nobody took the time or the interest to incorporate them into egg calcium signaling model. So there are thousands, well, maybe I'm exaggerating, but many calcium signaling models for other cell types, but not for eggs. So this quest for the best, for the appropriate model for calcium signaling X is very important because this is going to open again the avenue for day 1 diagnostic tool, non-invasive diagnostic tool for IVF. And the model works qualitatively because Carl really wanted to see these oscillations. We got the oscillations, but then the last meeting with the last of the students in that row said we need to calibrate quantitatively, right? So this, you should take a message out of this. And maybe somebody say, why are you being so open about things progressing so slow? Academia is slow, it's the long game, as Thomas said. You can keep doing projects and not progressing fast enough. For example, this one will have progressed much faster if we got funding for it. And we haven't as yet. So it relies on, working with MSc students. We also had an MPhil student, et cetera, et cetera. So this is like a bit of a struggle we have in academia to keep finding the resources and the people to continue work with on the problems we are interested in. So one thing we know how to do to improve the model further is to couple with mechanics, as I said originally. I've done quite a bit of work on that. So again, that's luck, right? Because I was working on this calcium and mechanics theme before I joined Cartif, just before. And then this is a kind of technology that we want to bring into this egg problem as well. This is from a different challenge. I'm just mentioning just to see how things mathematically can relate very well, but come from different biological challenges. So this is much later in embryogenesis when the neural tube is closing. So you have these calcium flashes, which are oscillations again, and they are autonomous. And they found out that if this calcium flashing does not work well, the choreography breaks down and the neural tube does not close. But from that, you can get these continuum models. And this is exactly what we need to apply also for our egg problem. So imagine this is not the neural tube, the neural plate is just the egg. And then you have, again, these calcium waves spreading. This is just a demonstrative slide. So we need to work with that idea of coupling calcium with mechanics. And then finally, I will finish. How much time do I have? I wasn't tracking the time. Yes, I will finish now. We did this GW4 network. As Thomas said, last year, we were thinking, how can we take this IVF work to the next level and get more people on board, collaborate with more people. Both of us love collaborating with people. So we said, why don't we apply for this GW4 Alliance funding? GW4 is Bristol, Bath, Cardiff, and Exeter. We did that, and our mandate is to improve the stagnating IVF success rates through maths and AI. We put AI there even though we're not experts, because other people are interested in it. And they can come and help and do their own projects, but it will still be all working towards the same aim. And we killed ourselves over creating 3 events since September, but they were very rewarding because we had 2 co-creation networking events in person. We had an online hackathon, and we engaged more than 90 early career researchers and IVF experts. We have an advisory board. We are finishing the project end of April. You can go to this website and see further what we are about and about the network. And of course, there is a link there if you want to join us. Some shameless promotion because we are looking to expand this community. And I will finish with some nice pictures. I like pictures and visualization. So that's from the final event. You can see here people working together in a table. We had teams from the hackathon presenting their results, early career researchers. Many people listed many IVF challenges, and we had that illustrator I mentioned, Thomas, before you joined. We were talking about Maria's great drawings, and I said we paid a lot of money to have an illustrator there in the event, which I think is the nice bridge to lead back to Thomas to tell us a bit more about those drawings and what is the challenges of IVF we have to face for the future. That's you, and I have to stop sharing.
Share this slide then. Because as you say, you know, I'd always been a fan of Maria's work, and let me share the screen then. There we go. There we go. And let me change that to normal. Is it? I have too many screens. This is the problem.
Yeah, what's happening there? Is that screen on screen or?
There we go. So hopefully you can see, this was from Elena. And what I would say here is that Elena isn't a scientist. She's an illustrator, a whole different set of skills than I ever will have. But one thing with running an event, an activity, Katerina and I found that although we brought all these people together, we weren't getting the time to talk to them. So what Elena was really invaluable at was taking out these key ideas, these big ideas, because although she may not know what a PIP2 is, a PLC zeta is, she doesn't care. She's looking for those big highlighted ideas that she can illustrate in such an amazing way. And so I was so struck by her work. And not beyond that, it's just a great reminder of the day. You know, Katerina has these photos of everyone working and looking and talking everything, but this is a keepsake that everyone wants to look at. You know, no one wants to see your conference photos, but this draws people in. This it excites people. This gets people to want to know what you're talking about. As I say, I was so excited by her work that we actually asked her back to the second event, where she did a second drawing of all the ideas that was going on. And I would say that, you know, this comes right back to Maria's work. So I wrote a piece for the SMB newsletter a couple of issues ago, where I was highlighting the use of illustrators and saying, you know, if you can do it yourself, great, wonderful, do it more. The first time you do it, you'll be terrible, but that's the same with anything. Do it more, you'll become better and better and better. If you have money, think about getting a professional to do it. It's worth the money because it gives you such a great keepsake for afterwards to remind you of all those ideas that can then spin off into the next 7 years of your life. With that, I think we're done. So any questions?
Well, I want to say thank you for, I keep saying it's me, but look at that. This is like beautiful illustrations. I feel like I'm always scribbling. I mean, one day I hope if I have enough time, I'll improve. Like you said, illustrating is another skill in itself, and it requires a lot more effort. Just learning how to do things on the tablet took a little.
But I would say doing it whilst listening to a set of scientific talks, I have enough trouble paying attention, let alone being able to draw at the same time. Absolutely incredible.
Well, thank you. And these illustrations are beautiful, but thank you. This was a wonderful talk. I didn't want to jump in, but you kept on mentioning my name. I had to at least thank you both. This was wonderful.
Wonderful. No, I mean, it's been a wonderful, wonderful talk, and it's been lovely to see how it's all come together. And I think there's a lot of lessons for us there about luck and hanging on in there and not giving up and taking your chances. Do people have questions?
Well, let's talk about that. So it's knowing when to give up. I would say it's not about necessarily giving up, but it's about following. One thing that Katerina is, where I love working with Katerina, she's very good at finding the money. And so whereas I want to know an idea, I want to do this piece of work, I want to, no, this is where the money is. Follow this direction. It will get us some money that we can do work with. And so that's one thing that Katerina does far better than I can do. You know, she's really good at that. So don't necessarily throw away ideas. I've got loads of documents where I put down ideas, but it's not necessarily let them lie and focus where the money is so you can do that next bit of work.
So I clearly could happily chat to these guys for a while, but please, does anybody have any questions? And it doesn't have to be about the science. It can be about anything. That's the point about these.
Yeah, I have a question, and it's still about the science. Since calcium waves were so emphasized here, and they have important role, I'm wondering about ion channels. Works, and I will just quote a person by the name Eisenberg, who ran a course a few years ago at the Fields Institute all about ion channels and calcium. And if you are aware of it, good, and if not, it's my little contribution.
You're absolutely right. I mean, it just takes you that lower down level of working with the channels then. So we didn't go into sort of the models that we've been using, but we certainly do have a model where Katerina will be explaining, so she knows a bit more about that model, but it has 3 channels that can be opened or closed in different orientations, and then that helps produce more calcium release. So Katerina, do you want to speak about that?
About calcium signaling models, yeah, maybe I should share my presentation again, because it would help me to again visualize.
So whilst Katerina's doing that, what there seems to be, and this was sort of the sort of dichotomy of calcium models, is that there seems to be sort of 2 general different groups. One is these ion channel ways of doing it, and then the other is this induced calcium release through the emptying of the endothelium. It's called endoplasmic reticulum.
Yes.
That's the one. And Carl doesn't like those emptying models because he says that there's so much in there, it never gets emptied. So we were trying to go more towards one of these ion models. So I'll leave it there for Katerina.
So do you see my screen, or do you see?
Oh, we can see the pictures at the end.
We saw the model previously. That's perfect.
Oh, I was showing the wrong slide. Sorry. Is this one I want to show?
Yeah, that's what we were looking at. We were looking at that.
Yeah. Oh, you were looking at that. Okay.
Yeah, we had that one.
Yeah, so endoplasmic reticulum is coming back to Thomas' original model. It's actually not a pool like that. It's just like a tentacle thing spread around the cytosol. Then you have on the ER these channels, and they cooperatively with calcium and IP3 open up and can release calcium, and that leads also to the stochastic calcium signaling, because many people are working on this stochastically because these are stochastic events. So this is something we have not even touched here. Thomas, you already covered the fact that some models assume the ER is emptying, other ones there is not, etc., etc. You may also get calcium from the outside, but the premise everywhere is that you have a series of fluxes. So calcium is coming from various routes. And you also have diffusion, and then this is buffer diffusion, because you have all these structures inside the cytosol that make the calcium not move as fast as it would in a homogeneous medium. And the crux of thing is that you have this calcium-induced calcium release, which basically is a nonlinear process that if the calcium concentration exceeds a certain threshold, then it stops. And this directly leads to those experiments that Carl wanted us to put in the model, and we did. which gives you exactly quantifies how calcium and IP3 work together to initially excite and open the channels and then close them. So I don't know if that answers the question. I mean, that's more about the how
Well, there was no
signaling models
question
work.
here. There was no specific question, just pointing to this Professor Eisenberg who ran the course, and at least one student was working closely with him, and the name is, the full name is Nathan or Nate Gold, who was at York University, probably finished his PhD by now, hopefully. And if anything of his work or other people from that course is relevant to you and help,
Thank you.
then
Yeah.
if not, okay, so I just verbalized names.
Yeah.
Good.
It's fantastic. Yeah.
I was gonna say maybe Dr. Halevi, can you type those names in the chat? Nathan Cole, I think I got, but I didn't. Heisenberg was the other guy, but if you can type them in, then we can make sure.
Well, Nathan Gold from York University worked with Professor Eisenberg, who was from Purdue or somewhere there in the US. And if I will catch that PhD or whatever, I'll communicate directly to you, Katerina, or to Thomas. And at the moment, I'm just finishing my shaving and getting
I think this could be,
so it's a cool morning here.
I think this could
snow,
be,
and
if I interrupt,
wish you all
is
the
this
best, guys.
just let me check. I suspect this is the person you mean. Let me just see. How do I do this? I'm always very so I don't know if you recognize him, but is this the person you meant? Do you think?
Should I stop sharing? I have to stop sharing, right, Katerina?
Okay.
Yeah, so now you know
Nathan Gold, right.
Yeah, that's him. That's him.
Yeah, perfect. Okay.
Very nice.
Very
Okay.
nice, friendly fellow.
Okay, so we that's now in the chat so everyone can see. So Isabel has some questions as well. They're wonderful questions. I could read them out, but actually it's to you, Thomas and Katerina. Maybe you read them yourselves and see what aspects you wish to comment on.
So it's all about timescale. So the question is, have I modeled the PLC zeta initiation? No, I haven't. Good question. Certainly something to be done. And what we were taking was the idea that you had been initiated, so it was just the interaction between the calcium and the PIP2. But so the reason why we're doing that is this idea here. So the sperm hits, however long this is, and then about 3/4 of an hour later, that's when the oscillations occur. So there is certainly a definite amount of time that you need for this PLC zeta to kick everything off. It's not an immediate wow, bang. But beyond that, I can't tell you in terms of its diffusivity. I don't know.
Do you wish to follow up again, Isabel, with this? Do you want to take it further in the chat? Okay, perfect. Anybody else have anything they want to talk about? Oh, yep, we've got another one. This is about there are only single spikes observed, but in the experiments there are oscillations. What do you think might be missing from the model in this case?
Right. Should I take it, Thomas?
Please do.
Yeah, so as I very briefly mentioned, you see oscillations because when there are these waves, right? So this is spatial temporal objects. So there is change both in space and time. But if you just fix yourself in space, you're only gonna see the time series, right? And that's why you see these calcium oscillations evolving in time, up and down. So it's part of the same story. Oscillations are just the fixed location version of a wave. So having said that, I think your question is important because it's that experimental capturing of the oscillations needs to be good enough to reproduce the wave, right? So exactly because we are sampling spatially, we cannot cover all aspects of the wave and measure all aspects of the wave. So basically a model that will create a wave is filling in gaps that the experiment is not giving. So it's important, this question between oscillations and wave is important. Thank you for that.
Do you have more? So, I would just follow up with, I mean, it's come up a couple of times, and certainly anybody in the UK is probably very concerned with this, but the idea of finding financing and how the search for funding sort of in some sense determines what you can actually do. And so for the sort of younger career researchers here, do you want to comment on that aspect of research, if you like?
Yes, we could. So basically, you have to be prepared for many rejections and consider that in game. So you keep applying. You don't be afraid to apply for small things as well, because it might lead to bigger things. That's, I think, the second takeaway I would relate to somebody.
And I actually agree with that. That's very much my strategy now, is that, you know, you can go for an EPSRC £10 million grant that will take you 6 months to write and la 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 la, or I could just go for something 10,000 and do a bit, and that's where I'm more aiming at, because if I don't get it, oh well, I've spent 2 weeks on it, and I can reuse it to get the next 10,000. I don't have to rewrite a whole 10-page document. Different strokes for different folks. I'm not saying one is better than the other. It's just my personality, the way I work. I'd rather get small pots of money that allow me to do little bits each time than a big, because be honest, I'm a dilettante. I will work on any problem that's interesting. So I don't have a £10 million overview question. It's not how I work. I have a 10,000, 20,000, 50,000 pound question.
Mm-hmm. Mm-hmm.
Yeah.
I think yeah. No, I was just going to say, I think it's very important to realize there are multiple ways to go about things. And I think that certainly you get the idea that you have to have your fellowship idea, or you have to, certainly as you're coming to the end of, say, your postdoc, oh, I have to have my fellowship now. I have to know what my big thing is. But actually you can construct what we see from your work is it's a long running program of work, but it's not been funded necessarily from the big pot of money. It's been put together in a very different way.
Yeah.
So I think it's quite good to see that you can do that. Is there anything else anybody would like to comment on?
I would like, should I say, Karin, that everyone wants that 5 million grant, right? I mean, that's like the Holy Grail, or maybe even 1 million, but yeah, you keep going and do something you enjoy. I think that's very important.
I think the enjoyment is very important. I mean, come on, that's why we're all here. I mean, we could be going and working in banks or something and earning lots of money.
For the money.
We want to be doing stuff that's intellectually stimulating and that we find interesting and fun.
Although saying that, so let's talk about Tim. So Tim, our student who did that wonderful work, he actually went to work for Nationwide afterwards. And let this be, so I don't know if we'll end here, but let me sort of give a moral of a story. Yeah, I've got ideas.
We're going deep here, Thomas. We're going deep.
Yeah. So Tim was a great researcher, absolutely fantastic. And we had got one paper published, and we're working on the second for this DDM stuff. Sent it out, the reviews came back, and reviews 1 and 2, fine, things to change, things to update, great. Review 3, this is not the paper I expected to read, and here's why it's terrible. What's that as a review? That's not how it should be done. So I want to give that to, if you're whatever stage of the career you're at, it doesn't matter if you're a Nobel Prize winner or your first paper, you're going to get bad reviews. And it's all about how you approach them. That destroyed Tim's confidence in himself. Like I say, I did a lot of work trying to boost him back up, but I could immediately see he was always on the fence of, do I want to do academia? Do I want a job? That firmly pushed him over. Fine, we've lost a great researcher. He's happy now. He's doing some really nice, cool security stuff with Nationwide. As long as he's happy.
And mortgage modeling as well.
Yeah. But I just feel it was such a huge shame to lose him through that. He was pushed rather than pulled.
Yeah,
I think it's certainly never a bad thing to remind ourselves we need to be kind. I think kindness in academia is a undervalued quality. We can
Absolutely.
disagree very strongly with each other, but kindness should always be there. I think that's very good.
That's the whole point of this community. So we can see we're all in it together and we can help each other. Because I think, at least as a theoretician, I felt like I was always alone in biology. And the truth is, maybe others can hear us. We're not alone if we reach out, and we don't have to reach out to other experimentalists, but we could reach out to each other. So we're not always reinventing models, and we can learn from each other. And I think that's a big, huge step.
Absolutely, absolutely.
Okay, well, I think if nobody has anything else they